before anything, we have to know two things before going deeply in pharmacology. At the beginning of the lectures, we have discussed the pharmacokinetic, and we said that pharmacokinetic is and a processes of four things.
Metabolism.
Yes. So here we have the pharmacodynamics, which is what we have in after the processes of the pharmacokinetic, we come to the pharmacodynamic. One comes after the other. أو وحدة مكملة للأخرى. So pharmacodynamics, that means it describes the actions of a drug or a drug on body and the influence of a drug concentration on the magnitude of the response. What do we mean by this? That means when the drug is absorbed and distributed, what is going to happen after that? رأسا شنو فائدة أني عندي drug absorbed and distributed؟ حتى إيش أحصل من وراء؟ تمام؟ So this is the pharmacodynamics. So after when you have a dose of certain drug, and that drug is going to be distributed, absorbed and distributed, it should reach the site of action. And when it reaches the site of action, it should produce or give physiological, إذا هو مثلا موجود normal عندي، وإذا أنا أحتاج فد شيء treatment, it should give the pharmacological effect. So study of the effect of the drug on the body is related to what we call it pharmacokinetic. So kinetic absorption distribution, and when the drug concentration reaches to the site of action, the pharmacodynamic effect will be produced أو خلينا نقول started in this case. The magnitude or the amount of response to the drug is dependent on the concentration of the drug at the site of action. As long as we have more absorption, more distribution, or good absorption, خلينا نقولها, good distribution, and we gain a good concentration of a drug at the target site or at the effector organ, we are going to have the pharmacological action. So the magnitude depends on the concentration of the drug reaching to the site of action. So starting with the kinetic and continuing with the dynamic. So this is what we have in dynamic. Most drugs exert their effect. It could be beneficial, and this beneficial effect when a therapeutic action is required. Beneficial effect, of course, it is the reason why what we give the drug or the therapy. And طبعا in therapeutic doses, of course, it is something studied. And when we have a high dose or some problem in the absorption or kinetic of the drug, we could get the harmful effect. Anything could be harmful, anything, even water, by interacting with receptors. So drugs may exert their effect, whether it is beneficial or harmful, by interacting with certain kind of molecules. نسميهم receptors. So these receptors are specialized target macro molecule. So they are huge by themselves. So they are macromolecule present on the cell surface mostly or within the cell. So it could be on the cell membrane or within the cell. We have very important thing to be determined in drug receptor interaction. Of course, we said that any drug to be effective, it should have a certain kind of receptor, whether we have a beneficial effect or harmful effect. So what is going to happen when the drug interact with the receptor? What is going to happen? طبعا الدرug هنا راح نسميه يكون على شكل signal, and if that signal is going to activate this receptor, what is going to happen later? How this signal is going to be transmitted تنقل inside the cell to give the action. We are going to see here. So this is what we call it signal transduction. So drugs act as a signaling molecule, and the receptors are signal detector. So the signal is the drug and the receptor is signal detector. The drug is termed here in pharmacology as an agonist. فagonist, a drug, a molecule that can bind to receptor and give the action. This is the agonist. So this is what we call it the agonist, أو نسميناه signaling molecule. If it binds to a site or receptor protein and activate it to initiate a series of reactions, فإذا drug receptor صار بيهم interaction. We have what here? Series of reaction, cascade of different processes. شنو cascade يعني؟ مثل سلسلة أو مثل الشلال، وحدة بعد الأخرى. Cascade of reactions, and this cascade will initiate a series of reactions that ultimately نهائيا result in a specific intracellular response. We get the response after all these processes. Here we end the series of chemical reaction. We end with what we call it an effector molecule. This effector molecule called second messenger. Okay? فهسه عندي drug receptor interaction, series of chemical reaction ending with the effector molecule or the second messenger. And these second messenger are part of the cascade of events. Cascade of events that translate agonist signal into the cellular or into a cellular response. فإذا انتقل عندي signal from outside of the cell to inside the cell, and we get, for example, مثلا عندي blood vessel vasodilator, I will get vasodilatation. مثلا عندي smooth muscle contraction, يصير بيها contraction on acting on certain kind of receptor, and as such as we are going to see later when we go deeply in autonomic nervous system. So we have here agonist, receptor, and second messenger signal effector. Coming to drug receptor complex, the magnitude قيمة أو something measurable of the cellular response is proportional to the number of drug receptor complexes. As long as we have more drug bind to the receptor, we get more action, وكل ما قل عندي interaction with the receptor راح يقل عندي response. So the magnitude depending on the number of drug receptor complexes. Okay, so this is very important. Coming to receptor state. Receptors in the body are not existing in one form. It is existed in two forms mainly, which is the inactive form or the R or the stable form, and the active form are star. And these two states of receptors are existing in equilibrium. Okay, activation and activation, activation and activation. So always exist in equilibrium that are reversible equilibrium with one another, usually favoring the inactive state. Of course, anything in nature favor the stability. So stability دائما تكون in the inactive state. Binding of the agonist مثل ما شفنا قبل شوية بالفيجر اللي سابق. Binding of the agonist causes this equilibrium to shift from the inactive state R to the active state. So binding of the agonist will cause shift of this equilibrium to the active state. صار عندي receptors أكثر شيء active. بينما normally without activation the inactive form will be more. And therefore we will get the pharmacological action or the biological effect إذا عندي إيش. We have another term called the antagonist. Agonist, antagonist. Antagonist, any drugs that bind to the receptors but do not increase the fraction of the active form of the receptor. So they favor the inactive form. They favor the inactive form. يعني إذا أنا عندي agonist وسوالي favoring to the active form أنطي الانتاغونست بعده, it will reverse the action of the agonist by favoring the inactive state of the receptor or stabilized form. Some drugs are called partial agonist. What do we mean by partial agonist? هسه أخذنا two things, agonist and antagonist. شنو partial agonist؟
يعني equilibrium بينهم؟
نعم. يعني شلون in terms إذا تفسر لي إياها in terms of receptor active and inactive. Yes.
يعني حيتساوي نفس الاكتف والاناكتف، ما راح يكون اعلى تنشيط الاكتف.
اي يعني شنو اعلى بعد شويه اكثر؟ نعم.
راح يزيد الاكتف بس باقل نسبه من
Very good. So it يعني إذا الأجونست راح ينطيني maximum effect يعني in terms of response معناه maximum activation of the receptor, partial agonist will give what? Less magnitude of less magnitude. راح مثلا إذا عندي مثلا smooth muscle contraction for example effect وصل إلى 100, partial agonist ينطيني مثلا 75%. اوكي؟ ما راح ينطيني كامل، ما راح اقدر احصل على الماكسيمم ثيرابيوتيك افكت. او مثلا in terms of physiology اللي اخذته المسل كونتراكشن فور اكزامبل بيها شنو؟ احنا قلنا threshold، نوصل للماكسيمم ليفل اوف كونتراكشن، ما راح اوصل الى الثريشولد، راح اوصل اقل من عنده ثلثه ربعه نصه وهكذا. سو هسه صار عندنا مصطلحات agonist, antagonist, partial agonist and receptors و signal effector molecule, second messenger. Okay, keep these in your mind, very important. If we can represent what we have talking about. For example, we have the drug as an agonist. It binds to the receptor, and we said that this receptor in the active form and equilibrium receptor, active and inactive, they are existed in an equilibrium. When we have agonist, it will favor what? The active form, and we get a response. And if we have antagonist, it will favor what? The inactive form or the R state only, not the AR star. And in this case, we get no response or very limited or reversing the action of the agonist. The partial, it is between these two effects. Coming to an important things, which are the types of receptors. Are these receptors or what kind, what types of receptors do we have in our body? Pharmacology, pharmacology defines a receptor as any molecule. قلنا هاي الموليكيول شنو تكون؟ macro كبير. So any biological molecule to which a drug binds and بس فقط bind, it should produce an action. A measurable response. يعني that effect should be measured. مثلا anti hypertensive drugs. اقدر اقيس البلاد بريشر. انتي اريثميك دراج من الاي سي جي اقدر اشوف ايش صار عندي changes بالاريثميا وهكذا. So I should have a measurable response so that I can determine my drug, how does it act, how does it work, and as such. These receptors in our body may be divided into 4 families, and every family may have members. ممكن أن تتقسم. فمن أقول family يعني هاي مجموعة وهاي مجموعة وهاي مجموعة. Receptors, not receptor. So I have 4 families of receptors. The first one called ligand-gated ion channel. Ligand-gated ion channels, or between 2 brackets, أسميهم ionotropic receptors. لا تصفنون بوجهي اكتبوها. إذا أعطيكم نص اكتبوا. ضبطوا إنهم قاعدين ماشيين. So, ligand-gated ion channel called ionotropic receptors. Tropic, that means triggered or stimulated. Tropic or trophic, that means triggered or stimulated. So I have ligand-gated ion channel. This is the first family. Second family, I have G protein coupled receptors. G protein coupled receptor. ثانية أسميها between 2 brackets metabotropic receptors. Metabo from metabolism, metabotropic receptors. The third one is what we call it enzyme-linked receptors. بين قوسين example خلو tyrosine kinase receptors. Tyrosine kinase receptors. And the fourth family is intracellular receptors. The first 3 are extracellular. The last one only intracellular. هاي فقط اللي عندي. كل الريسبتورز اللي راح ناخذها could be grouped in one of these 4 families, or could be allocated. Coming to what we have talked about, to explain it on a figure. So the first one, سميناها أول وحدة
ionotropic receptor.
ionotropic receptors. شنو معناها ionotropic? What do we mean by this? Yes. So I have here a channel, and this channel will cross the phosphodilated membrane. And every channel of these types of receptors consists from 5 subunits arranged around a hole. So 5 protein subunits arranged around a hole, okay? And permit the passage of certain kind of ion, either cation or anion, according to the type of receptor. For example, we have nicotinic receptor that permit the passage of sodium. بينما البنزودازepine receptors أسميها chloride channel permit the passage of chloride. من تجون على central nervous system تاخذ. So till now we have example nicotinic receptor sodium channel permits the passage of sodium, and let sodium enter inside the cell after the drug has been bind on a specific site on that receptor, making the channel to be opened, and therefore more sodium will enter inside the cell. نعرف from our study in physiology that the cell membrane is existing in polarized form. تمام؟ الممبرين مالتنا polarized. Positive from outside, from the extracellular site, and negative from the intracellular site. When you have ions or sodium ion enter inside the cell, what is going to happen to the intracellular site charges? What is going to happen?
تزداد
يعني ايش صار عندي اسميها الحالة؟
دخل الصوديوم
إيه.
بدأ تحسن موجب ولا تعادل؟
لا مو تعادل، ماكو تعادل عندنا. إيه. it becomes more positive. هاي شو اسميها الحالة؟ تحول الشحنة إلى شحنة أخرى. Depolarization. Depolarization معناها activation or inhibition. بهالحالة أنا الممبرين normally عندي positive from outside, negative from inside. من دا يصير بي depolarization. إيش دا يصير بيها السيل بهالحالة؟ تحول من الستيبل إلى الأكتف. تمام؟ So depolarization, keep in your mind. خلوا ملاحظة يلكم. Depolarization is activation. So this will cause change in membrane potential or ionic concentration within the cell. من صار عندي تغير بالممبرين potential يصير عندي activation, many enzymes, many molecules, other channels, potassium channel, calcium channel وغيرها, ويصير عندي intracellular effect يظهر. طبعا هذا الاكشن راح نشوفه is very rapid. هل اكو يعني اللي دا اشوفه بالفيجر اكو عندي هنا second messenger او effector molecule موجوده عندي اللي حكينا عليها. عندي cascade of reactions حتى احصل على الافكت معي. تمام؟ فاذا الاكشن here is so rapid. So when I have an excitable tissues, like شنو excitable tissues الموجودة عندي؟ neurons والmuscle, muscular tissues. So they are excitable tissues. Wherever I need rapid action, so I have شنو عندي؟ ionic channel. Or I have ionotropic receptors, as we are going to see later. Coming to the G protein coupled receptors. What do we mean by G protein? G protein is a protein consisting from 3 subunits, alpha, beta, gamma, and this G protein is linked to the receptor. But when it is going to be activated, once the drug is bound to the receptor, this molecule or G protein is going to bind with the receptor, and then the action will be produced as we are going to see بعد شوي. Protein causing, for example, protein phosphorylation. هنا بدنا نشوف بدنا يصير phosphorylation ورياكشنز معناها عندي هنا second messenger. تمام؟ فإذا ليش سميت الجي بروتين coupled receptors metabotropic receptors? Because I have a cascade of several metabolic processes until I will get the effector or the second messenger so that the intracellular effect will appear after that. طبعا راح تستغرق لي وقت معين اطول من اللي استغرقته الionotropic receptors and within seconds. The third type of family is called enzyme-linked. شنو معنى enzyme-linked? These receptors containing within itself, within the molecule, enzymatic activity. يعني الريسبتورز بيها انزيم مو مثل الجي بروتين، جي بروتين مولكيول منفصله عنها، يصير ارتباط بعد ما يرتبط الدرج بالريسبتور. Here the receptor has enzymatic activity بيها انزيم معين. And this enzyme going to produce or phosphorylate itself نسميه O2 phosphorylation. When the drug is going to bind to the receptor, autophosphorylation or activation of the receptor by itself ما احتاجت إلى molecule خارجية وسوت لي activation. هي receptors ب tyrosine kinase for example activity. بعدها أيضا راح يصير عندي several metabolic processes وأيضا راح أحصل على second messenger and we have an effect in this case. The fourth type, which is the intracellular receptors, here I don't have anything on the cell membrane. In fact, the drug should enter inside the cell. ونعرف درسنا what are the factors affecting the passage of any molecule or a drug inside the cell. شنو الفاكتورز اللي تأثر عليهم؟ blood flow, lipid solubility, يعني البرفيوجن، cardiac output وهكذا. كل هاي الفاكتورز راح تجيني هنا تبدي تشتغل where the drug is going to be يعني enter inside the cell. Of course, any lipid soluble drug is going to pass rapidly through the cell. Once the drug inside the cell, it will bind with a certain carrier molecule. Mainly, these carrier molecules are transcriptional factors. This carrier molecule will translocate drug molecule complex inside the nucleus. ايش عندي بالنيوكليوس؟ DNA RNA. شنو هذول؟ جينات. منو يأثر عليهم؟ transcriptional factors اللي هي ارتبط بها الدرج. الدرج in this case will activate the transcriptional factor. فايش راح يسوي لي الجين؟ يسوي جين transcription. بعد ما يصير عندي جين transcription I have messenger RNA بعدها translation إلى بروتين وهذا البروتين هو راح ينطيني الأكشن اللي هو could be an enzyme metabolic enzyme ممكن أن يكون receptor huge molecule يتحول إلى cell membrane ممكن أن يكون هرمون ممكن أن يكون cytokine involved in inflammatory processes وهكذا. So protein phosphorylation altered gene expression مثل ما قلنا gene expression I will get the intracellular effect. طبعا of course this process will take a long period of time. So here we have in the ionotropic time lapse to give the action will be here in millisecond while here will be in seconds while in the third and fourth type of receptors may take more than may take hours or even days. نرجع بعد أكثر نحكي على receptors. Considering ligand-gated ion channels or سميناها ionotropic receptors. Once the drug or bind to the receptor, ions will pass inside the cell. احنا قلنا البروسيس شنو؟ راح يزيد عندي positive charge inside the cell and we get depolarization. شنو معنى الهايبربولاريزيشن؟ What do we mean؟ وقلنا الديبولاريزيشن معناها شنو؟
تغيير
activation. What do we mean by hyperpolarization؟
يعني الاكتيف يكون عالي كلش؟
نعم.
التنشيط يكون كلش عالي؟ اكتيفيشن؟
اللي شنو؟
اللي هو تغيير البوزيتيف.
لا. بعد.
هو المواد زاد راح يصير عكس.
شنو هايبر؟ زيادة. بولاريزيشن شنو؟ قطبية عالية. زين، القطبية مال الممبرين شو تكون؟
راح تكون
بوزيتيف extracellular negative intracellular. إذا hyperpolarized membrane from inside شنو معناه؟
بوزيتيف تزداد.
positive no negative.
نيجاتيف عكس الديبولاريزيشن.
So it will be more negative.
عكس الديبولاريزيشن.
زين. إذا الممبرين more negative, is it easily to be activated?
لا تقلل على التنشيط.
إذا السيجنال أحتاج more signal to be activated. تمام؟ أحتاج more signal to depolarize the membrane. إذا I will get شنو؟ inhibition or activation. So إذا عندي الأيون مالتي أنايون مثل الكلورايد قلت لكم بالسنترال نيرفس سيستم نعرف البنزودايازابين مثل الدايازيبام وغيرها هي inhibitory تمام؟ central nervous system sedation relaxation muscle relaxation وهكذا. الكلورايد channel receptors أو البنزودايازيبين receptor أو GABA receptor نسميها تكون كلورايد channel فإذا they are inhibitory because they produce hyperpolarization. ليش؟ لأنه الأيون عندي أنايون كلورايد. On the other hand, on nicotinic receptors اللي هي موجودة عندي بالسكليتال ماسل والأجونست عليها هو الأسيتيلكولين ونعرف أنه from our study in physiology that acetylcholine is going to produce skeletal muscle contraction يعني activation. فإذا بهالحالة أحتاج إلى cation. So I will have in this case depolarization and I will get activation. G protein coupled receptors as the drug binds to the receptor, G protein is going to activate certain metabolic processes, generate second messenger, producing phosphorylation of certain kind of proteins to get cellular effect or increase intracellular calcium or other effect. المهم ان بالنهاية احصل على response نهائي. ما مهم هذا لانه بعدين راح ناخذها بتفاصيل بقية ممكن at the same time G protein receptor is going to activate or inhibit or زائد activation ناقص inhibition is going to activate or inhibit other channel protein existing on the cell membrane مثل calcium channel, potassium channel, sodium channels, and all these channels are voltage dependent, most of them. فكل ما صار عندي change بالممبرين potential أو تحفز عندي G protein ممكن أن يغير لي activity of the other ion channels like sodium or potassium or whatever, or calcium. For the kinase linked receptors, of course, we said that we have an enzyme here, and this enzyme auto activated or auto phosphorylated to get phosphorylation or gene transcription or protein synthesis, and we will get the effect. And the intracellular one is involved in gene transcription and then protein synthesis. Coming to transmembrane ligand ion channels. Here we have, as we said, or as we have discussed before, we have 5 subunits. 5 subunits arranged around a core. And this core permits the passage of the ion, whether they are anion or cation. Once the drug binds to the receptor, it will induce the opening of the channel and permit the passage of the ions inside the cell, producing maybe depolarization or hyperpolarization. These kind of channels having an extracellular site, which is the site for drug interaction, and intracellular site from which the ions can pass through. Stimulation of the, for example, nicotinic receptors by acetylcholine results in sodium influx, potassium outflux, generating an action potential in a neuron or contraction of the skeletal muscle. بعدين راح نجي عليها بالتفاصيل. The second one is the transmembrane G protein coupled receptors. Transmembrane G protein coupled receptors, if we look to this figure, we have an extracellular domain. Domain يعني مثل ما نقول مجال. عندنا مجالين للbinding of the drug with the receptor, والثاني يكون عندي within the membrane transmembrane, وعندي جزء داخلي inside the cell. So the extracellular site containing the site of binding of the drug to that receptor. The extracellular domain of this receptors contain ligand binding area, and the intracellular domain interact when activated. So once the drug binds to the receptor, it will produce conformational changes in that receptor, making it easily or can bind easily with the G protein اللي هو موجود قريب عليه. So once the receptor activated, G protein will bind to the receptor. We have many kinds of G proteins. We have different kinds of G protein. We have G protein S, G protein I, and G protein Q. G protein S is من S stimulator. That means it stimulates the activation and always mainly include G protein coupled receptors related to the activation of the second messenger اللي هو adenyl cyclase enzyme. Adenyl cyclase. Adenyl cyclase is an enzyme responsible for the production of cyclic AMP. دائما cyclic AMP يسوي لي phosphorylation، يسوي لي enzyme activation، ممكن ان يحفز لي calcium release from endoplasmic reticulum. It could act يعني differently يعني inside the cell. فهو يعتبر لي second messenger. Okay? فإذا if I have GS راح يمر علينا أنواع الريسبتورز. إذا it is related to the activation of adenyl cyclase. Adenyl cyclase أيضا مثل G protein يكون inactive by itself unless the receptors are going to be activated. Drug receptor interaction, activation of the G protein. G protein the active site. So the phosphorylation from GDP اللي هو guanosine diphosphate اللي هو inactive يتحول إلى triphosphate, and the triphosphate is going to phosphorylate inactive adenyl cyclase leading to active adenyl cyclase اللي بالنهاية توصل على cyclic AMP. cyclic AMP اللي هو مثل ما قلنا the second messenger. The second messenger or the effector molecule. What do we mean by GS, GI, or GQ? As we said, GS and is linked to the activation of adenyl cyclase. GI شنو معناها عكسها linked to inhibition of adenyl cyclase. زين بقى عندي GQ شنو؟ GQ is related to activation of phospholipase C. Okay? Not adenyl cyclase. For GSGI, activation or inhibition of adenyl cyclase. GQ is linked to the activation of phospholipase C, generating two second messengers, مو واحد فقط cyclic AMP. One of them is inositol triphosphate or 1,4,5 triphosphate, نسميه inositol triphosphate, or DAG اللي هو diacylglycerol. هذول اثنينهم second messenger. How many second messengers do I discussed till now? كم سكند مسنجر صار عندي؟ ثلاثة. شنو هم؟
الفوسفور
cyclic AMP, CAMP, IP3 and DART. Okay? So I have these three second messengers or kinds of second messenger. DART and cyclic AMP activate different protein kinases. شنو kinases بالبايوكيمستري؟ شو يسوي كاينيز؟ استيليز مثلا remove of the ester group. methylase removal of the deaminase شنو كاينيز؟ I know هو كلها تسبع reactions. But what is the action of kinases? Yes, phosphorylation. So DART and cyclic AMP activate different kind of kinases within the cell, leading to myriad linked physiological effect. If this is normal, if I have a certain abnormal condition, then I can give a drug to reorder disorder or to bring out the normal function again. Enzyme-linked receptors. These kinds of receptors possess tyrosine kinase activity. For example, عليها are the insulin receptors. مثال على tyrosine kinase أو enzyme-linked receptors are insulin receptors. So, as I said before to you, these receptors possess تحتوي tyrosine kinase activity as a part of their structure. يعني receptor هو أصلاً به enzymatic activity. When the peptide hormone, for example insulin, bind to two of its receptors, دائماً الانسولين bind to two subunits, not وحدة. Okay? So, these receptors, to be activated, it should bind to molecule of the agonist. When peptide hormone insulin bind to two of the receptor subunits, there is intrinsic tyrosine kinase activity. Intrinsic tyrosine kinase activity, causing phosphorylation of receptor itself. لأن الريسبترز ما تحتاج إلى فد شيء بعد molecule يمها أو قريبة عليه. In fact, we have autophosphorylation. Tyrosine kinase will cause phosphorylation of one unit, and the second unit will cause phosphorylation of the adjacent unit. This is what we call it autophosphorylation after binding of insulin to the receptor. Enter phosphorylated receptors. هسه صار فيها receptors بعدها صار فيها phosphorylation. So once the receptors are going to be phosphorylated, other peptides or proteins that subsequently activate other important cellular signals will be activated. For example, هنا أنا عندي insulin receptor substrate. Insulin receptor substrate will be tyrosine A1. Tyrosine of the receptor phosphorylated form will phosphorylate insulin receptor substrate, and I will get different effect of insulin. اللي هو increased glucose uptake by the cell, increased gluconeogenesis, increased glycolysis, generation of phospholipids, وهكذا different actions of glucose or insulin inside the cell. This response lasts on the order of minutes to hours. Of course, طبعا مثل ما قلنا قبل شوية.
long time.
Yes, it will consume a certain period of time, and most common enzyme-linked receptors هذا النوع تكون عندي for example اللي أخذتوها بالimmunology. We have many cytokines مثل tumor necrotic factor, epidermal growth factor, interleukin 1, interleukin 2, يعني different cytokines which are involved in our immunity have these kind of receptors. So epidermal growth factor, cytokine receptors, insulin, and others. So keep in your mind that these kind of receptors are mainly for cytokines, hormones like insulin and other things. Intracellular receptors these receptors entirely intracellular. I have no extra cellular domain, never. And therefore, the ligand must diffuse into the cell. طبعا الليجنز ممكن أن يكون agonist، ممكن أي molecule ترتبط بالريسبتور. أي واحد، أي شيء. agonist, antagonist, partial, whatever. فممكن أن أقول ligands أو agonist or whatever. So therefore ligands must diffuse into the cell, lipid soluble of course, to interact with the receptor. فإذا الريسبتور موجود عندي داخل الخلية as a carrier molecule. Binding with the agonist activate receptor via dissociation from a variety of binding protein. شنو معناه؟ الآن عندي many molecules inside the cell, many proteins. When the drug bind with a certain protein, جزء من هالبروتين راح يؤخذ ويا الدراج. يصير عندي drug protein complex. This complex then translocated inside the nucleus and will going to affect مثل ما قلنا شنو؟ gene transcription and generate messenger RNA بعدين translation إلى protein and we will get the effect in this case. Also, the effect will last more than from hours to days. Other targets of intracellular ligands ممكن أن يكون structural protein inside the cell، ممكن أن يكون enzyme، ممكن أن يكون RNA أو ribosome. وراح نشوف تمر علينا فارماكولوجي drugs that act intracellularly and having or their target راح يكون عندي ribosome أو proteins أو enzymes أو RNA يعني by itself. For example, we have a tubulin. سامعين بالتubulin. What do we mean by tubulin? شنو tubulin? Microtubules شنو؟ Microtubules. ايه شو تسوي؟ What do we mean by microtubules? لا. شلون تنقسم الخلية؟ شلون تنقسم الخلية؟ خلية شلون تنقسم؟ أنا أعرف؟ من تنقسم الخلية شو يصير بيها؟ لحظة الانقسام ضعف بعدين كروموسومات الضعف
ينقل الكروموسوم
شلون ينقل بيش؟
هو بالتيوبيول
يقول يا ابني ليش ما تحكي؟ ليش ساكتين؟ أول مرة هاي دا أسمع صوته هنا رجاء طلاب بناتي أولادي قلت لكم من تحكون بصوت ناصي ليه يأثر علي؟ واللي ياخذ يريد راحته خليه يطلع براحته. قلنا لكم غياب وما حد هذا اللي يجي مقتنع هو يريد يدرس ويريد يتعلم واللي ما يريد بكيفه جاي للونسه براحته. منو جابره؟ ايه كروموسومز تتضاعف ايه تمام وبعدين ايه بواسطه المايكروتيوبيولز او التيوبيولين اللي هي اساسها polymer of these will form the microtubules. زين. فإذا tubulin is a target of many anti-neoplastic drug أو نسميها anti-cancer drug. فهنا الريسبتر مالتي صار هاي الموليكيول فقط. ما عندي لا ريسبتر ولا عندي نيوكليوس ولا عندي أي شيء. شنو عندي؟ بس المايكروتيوبولز. رأسا يجي الانتي نيوبلاستيك ايجنت او فور اكزامبل باكليتاكسين او انتي كانسر راح يرتبط بالتيوبولين يمنع البوليمرايزيشن مالته، يمنع فورميشن اوف مايكروتيوبولز، يمنع انقسام الخلايا. خلاص. اذا هنا الريسبتر مالتي شنو صار؟ التيوبولين اللي هو بروتين انسايد ذا سيل، مثل ما قلنا ستراكشرال بروتين موجود داخل الخلية. او ممكن ان يكون انزيم. For example, dihydrofolate reductase enzyme. Dihydrofolate reductase enzyme ايضا موجود بالانتي كانسر دراج والانتي مايكروبيال ايجنت مثل الترايميثوبرين اللي ناخذها تابلت سلفاميثوكسازول اند ترايميثوبرين التابلت. طبعا نعرف هاي they are antibacterial agent. Trimethoprim is a dihydrofolate reductase. وين موجود؟ موجود داخل الخلية. فهنا التارجت مالتي inside the cell dihydrofolate reductase. ما عندي ريسبتر هالحالة. اوكي؟ زين. Also antibacterial agent. راح ناخذها بعد المحاضرات الجاية. Bacteria has 5 or 50s subunits. طبعا البكتيريا بيها subunit للرايبوسوم. 50s بعدين نجي على تفاصيلها. 50s subunit of the bacteria ribosome is a target of macrolide. Azithromycin, erythromycin, erythromycin. They are target for 50s. وين موجود ال 50s رايبوسوم؟ ايضا بداخل الخلية. فهنا أنا أيضا ما عندي ريسبتر حتى كلها هاي ممكن أن تشكل لي intracellular molecules or intracellular target for my intracellular acting drug. فإذا it could be enzyme or molecules or RNA or ribosome. Of course, when drug bind to the receptor, it will give a signal, very small signal. How these signals is going to produce the effect? Of course, there must be a signal amplification. That means the ability to amplify small signals اللي هي فقط إشارة من الدرج وصار بي الميتابوليك بروسيسز وتحفزت عندي السكند ماسنجر وغيرها من الامور. So I have a signal amplification. I have an important thing also considering drug receptor interaction. We have a term called desensitization or downregulation. When you continuously give or administer the agonist for a long period of time. These receptors are not going to be activated anymore. Why? Or I have a reduced response after continuous administration. Why? Because the receptors are either desensitized, صار بيها يعني تغير معين chemical reactions بحيث تكون non-responsive to that agonist again, or they may be down-regulated. طبعا احنا what do we mean by down-regulated؟ يعني عددها يقل. شلون يقل عددها الريسبتورز؟ انا عندي مثلا سيل وبيها ريسبتور معينه على السيل ممبرين. شلون يقل عددها ممكن ان يقل؟
يعني قد ما ارتبطت بعد ما كان
لا هذا يصير بالبداية وبعدين يصير نتيجة what you said I have down regulation. شلون قل عندي العدد مالتها؟ ايش دا يصير فيها ودا يقل عددها؟ اي شلون دا يقل عددها؟ يعني هي اصلا عدد مثلا محدد عندي انا ودا انطي continuously agonist activation. بعد ما تشوف السيل يعني activated continuously hyper activation بعد ما تحتاج تبدي تسوي يعني مثل ما نقول some maneuvering to protect itself. ايش راح يصير عندي بالنمبر اوف receptors؟ قلنا النمبر اوف receptors راح يقل. How this is going to happen؟ شو يصير؟ هو شنو عبارة عن قلنا ريسبتر شنو عبارة عن شنو؟ بروتين. منين يجيني البروتين؟ تمام. إذا وحدة من الطرق after continuous activation وهذا ايش دا يصير عندي؟ يقل عندي transcription of that protein. So their expression on the cell membrane will be lowered in this case. هاي وحدة منها. Or sometimes the receptor is going to be internalized so that no more binding site is going to be exposed on the cell membrane and therefore there is no enough number of receptors that could bind with the drug. Okay? فهاي نسميها اما desensitized انه بعد النمبر ما يتغير بس ما تتحفز بعد anymore. Some chemical changes will happen, or they are down regulated. Number of these receptors are going to be reduced. وهاي نشوفها بالaddiction and addicted people when they continuously use certain narcotic drugs or drugs hallucinogenic or whatever. They need after a period of time to increase the dose in order to get the same effect. Why? Because we have desensitization or down regulation of receptors. قل عندي receptor لذلك احتاج agonist اكثر حتى احفز اكثر. نكمل بالمحاضرة.